For this activity, we're going to need a microscope, microscope slide and cover slip, an onion, scalpel, forceps and a white tile to act as a cutting board, paper towels, a dropper bottle of iodine and a little bit of distilled water. First thing we're going to do is head to the teacher's desk and cut a small square from our onion. It needs to be roughly 1 centimetre in each direction. Your teacher may decide to help you with this step and provide you with a pre-cut section of onion. Back on our own bench now, we need to lay out a paper towel to work on. This helps us to see the glass slides better and helps us avoid staining the bench if we overdo the iodine later on. Now comes the tricky bit. The part of the onion piece that we want is a thin membrane that separates the layers. Using the forceps, we need to gently peel away the layer. Once it starts to come away, you should be able to peel it off in one piece. Before we place it on our slide, we need to add a small drop of water and place our membrane on top. Use the edge of a paper towel to soak up any excess water. Next, we need to add a stain to make the cells easier to see. In this case, we are using iodine. Add a couple of drops to the top of the sample. To finish our slide, we need to add a cover slip. To avoid getting air bubbles under the slip, we need to place one edge on the slide and then lower the other side down like a hinge. Here's that cover slip move from a different angle. Using the edge of a paper towel, we can soak up any excess iodine, and then that's it. Our slide is ready to go on the microscope. If everything has gone to plan, then these are the sort of images you can expect to see. At 40 times magnification, you can start to see the individual cells marked out by the cell walls. At 100 times magnification, the individual cells are much clearer and you can start to see some of the cell's nuclei. At 400 times magnification, we can see right into a single cell or two. Here you can see quite a few of the organelles inside the cell.